you know, the information goes through the platform.
Okay.
So, Michael, me dá os 15 segundos, tá?
Ok. Estamos online, professor.
Perfeito. Bom dia a todos. Boa sexta-feira para todo mundo. É um prazer enorme recebê-los aqui em mais um ciclo, né, do nosso colóquio semanal aqui do Estúdio de Física de São Carlos. Ainda estamos no evento remoto, claro, e assim permaneceremos esse semestre com certeza e provavelmente o próximo, né? Antes de falar do palestrante de hoje, só dar algumas notícias, uns avisos, né? Lembrando que a gente sempre está espaçando, né, os colóquios em 2 semanas, né, para não sobrecarregar ninguém em relação às atividades virtuais. E o nosso próximo colóquio é dia 11 de junho, tá? Vai ser o doutor Heráclio Tavares, do IFUSP, foi uma sugestão da professora Cibele, e ele vai falar, né, de história e ensino de física num contexto aí sobre a visão do César Lattes. Então vai ser bastante interessante. Então já anotem aí na agenda. Nós faremos as divulgações, né, na semana do colóquio, mas já fica aí essa informação para vocês ficarem ligados aí no dia 11, não deixarem de participar. Tá? Eu vou fazer a transição para o inglês agora. So now, switching to English. It's my pleasure to be here with Giulio, Cerullo, and Paulo Miranda. Paulo Miranda suggested the name of Giulio to be given the talk today, and therefore it's my pleasure to invite Paulo Miranda to introduce Giulio in this colloquium. Paulo, please.
Okay, thank you, Gregorio. Thank you, Giulio, for accepting our invitation. I will just say a few words about Giulio, his career. He finished his undergraduate in electrical engineering at the Politecnico di Milano in Milan, Italy in 1988. And then he spent almost 2 years after graduation at a laser company in Germany. And then he went back to Milano, where he became a university researcher, probably what's equivalent of assistant professor in 1991. Then in 1995, he spent 1 year at the Lawrence Berkeley Laboratory working with Chuck Schenck. He's, for those that know ultrafast spectroscopy, Chuck Schenck is a father of the ultrafast spectroscopy, ultrafast lasers, and as a curiosity is the same supervisor of Carlos Brito Cruz, yes, which was president of FAPESP for many years. Then in 1999, Professor Cerullo became associate professor at the Politecnico, and in 2011, a full professor. His research lines involved just the generation of coherent and tunable ultrafast laser pulses. In that area, he is widely known as the inventor of the non-collinear optical parametric amplifier, which is a widely used device in research labs and in commercial equipment, okay? And he also uses these ultrafast pulses to do spectroscopy with extreme time resolution of a few femtoseconds, and that's the theme of his talk today. And he applies this to many different areas of condensed matter and biomolecules, specifically charge transfer and energy transfer in conjugated organic molecules, biomolecules, carbon nanotubes, and also coherent vibrational spectroscopy and dynamics, and also microscopy of polymers, proteins, dynamics of metal insulated transitions in highly correlated materials, 2D materials, just to mention a few. Okay, so wide range of applications. I just want to highlight one accomplishment of his, which is very fun to me. He was the first, he performed the first time resolved electron transfer from a polymer to a fullerene molecule, which is a prototypical system of organic photovoltaics. So he was the first one to tell us how long does it take for an electron to jump from a polymer to an acceptor molecule, okay? And this, and in 2011, he also was awarded the prestigious European Research Council Advanced Grant for developing two-dimensional electronic spectroscopy for biomolecules, okay? Another field of research is in the area of microfabrication with ultra-short laser pulses, where he has several important advances in waveguide and microfluidics fabrication, including the first demonstration of net gain in a laser-written waveguide with erbium-doped glass. He has obviously supervised many masters and PhD students and postdocs. I'm not even sure the correct number. Maybe he even doesn't know completely because so there are so many of them. And he has been an editor of optics letters and a chair of several important conferences such as CLIO Europe, Ultrafast Phenomena, International Conference on Raman Spectroscopy, and so on. He's a fellow of the Optical Society of America and of the European Physical Society, where he serves as a chair of the Quantum Electronics and Optics Division. He has more than 450 publications, with which received more than 22,000 citations. His H-index is 79, and with these numbers speak for themselves. So by now I should let Giulio Cerullo speak to us, okay? Thank you very much, Giulio, and please welcome to our virtual colloquium.
Yes, many thanks, Paolo, for your really kind introduction. Can you see my screen?
Yes.
Yes, so it was a really nice introduction, and actually I'm really pleased to hear the name of Professor Brito Cruz, because when I started doing my first ultrafast spectroscopy experiments in the mid-90s, I was reading all the papers by him with Chuck Schenk. So I was really studying in much detail what he has done, because he has really pioneered the field. And what I'm really doing is pretty much on the footsteps of his pioneering work on really using few-optical-cycle light pulses to investigate primary events in biomolecules. So this will be the topic of my talk, and this is my activity that I carry out at the physics department of Politecnico di Milano. And since I assume that the audience will be quite broad, I will start with a really general introduction to ultrafast optical spectroscopy, also with a bit of historical perspective. So how one could generate such short light pulses, and what is the history of using short light pulses to look at fast phenomena. And then I will move to the applications, and in particular, I will focus on some very fast processes in biomolecules that are mediated by so-called conical intersections. So I want to introduce you to this concept of conical intersection, which is a peculiar arrangement of energy levels in molecules, which I see like a door that allows for excited state, for a very fast transformation from one electronic state to another. And then I will show you how nature uses conical intersections to perform two functions. So one fascinating process is the primary event of vision. So I want to show you how one can use conical intersection to transduce the energy of a photon that strikes our retina into an electrical impulse, and how one can map in real time the dynamics of this process. And then, if I have time, I would like to show you how one can use these conical intersections in order to understand the photoprotection mechanism in DNA. So how the DNA molecules can effectively dissipate the absorbed sunlight without being damaged. And all this is achieved through ultrafast optical spectroscopy. So let me start with a really broad and general introduction to ultrafast optical spectroscopy. And I like to start always my presentation by showing this painting of 1821. This is a painting by the French painter Théodore Géricault that's called Les Derbies de Psaume. So this painting actually shows galloping horses, and the way the painters, you can find many such painters in the paintings in the 19th century, and this is how the painter was imagining the galloping phase of a horse. So our eye doesn't have enough time resolution to really resolve the moment when all the four legs are lifted from the ground. So the idea that the painter had was that when the horse is galloping, so when the four hooves are lifted from the ground, the legs are spread apart, are spread wide apart. Now, is this a correct picture or not? We don't have enough time resolution with our eyes to tell if this is a correct picture. So what we need to do is we need to shine an ultra short flash of light to freeze the motion of the horse and do an experiment that tells us whether this painting is correct or not. So, of course, every experiment needs a sponsor. And indeed, the sponsor of this, what is considered the first ultra-fast optical experiment, was Leland Stanford, who, of course, everybody knows Stanford University, that was founded by him. And so Leland Stanford was, of course, living in the West. He was very fond of horses. So he really made a bet with a friend. How would effectively the horse gallop? How would be the galloping phase of a horse? And so he gave a task to this photographer, Edward Muybridge, to develop a flash photography technique with a time resolution of about 1 millisecond, so 10 to the minus 3 seconds. So this was a quite long and adventurous process. You can Google the story of Edward Muybridge. It's very interesting. It's quite an interesting story, what happened to him before he could shoot these pictures. But in this very famous sequence of pictures in June 1878 on the Palo Alto racetrack, he could really freeze with these flashes of about 1 millisecond the motion of the horse and the galloping phase. And indeed, when you look at the phase when all the four legs are lifted from the ground, you see that you get now a picture that is completely different from the previous one. Now the legs are not spread apart, but they're rather close together. So having a flash of light that freezes the motion of the horse, we learn something about these dynamics. And indeed, you can put together these snapshots, these pictures, and make a sort of a slow motion movie of the galloping phase of the horse, and you can in this way retrieve the motion. This was done on many different processes, many different moving animals. Now this is flash photography with a time resolution of 1 millisecond. Of course, we want to look at faster processes, generate shorter light pulses. Now, if you look at a modern painting, this is a painting of the 20th century after the Muybridge photographs. And now the galloping horses, you see that this picture is correct. This is exactly how the horse gallops. So this is now feeding into painting this new knowledge that we have learned with this kind of optical spectroscopy. Now, how fast can you go? How fast events can you follow, and how short light pulses you can make? So another important development was achieved by Harold Edgerton at MIT, who developed stroboscopic photography, which probably most of you have seen this kind of pictures. This photography has a resolution on the order of microseconds, which is another 3 orders of magnitude shorter with respect to the previous Muybridge experiments. Now you can ask yourself, what time resolution would you need if you really want to look not at macroscopic objects, but at microscopic objects, such as molecules moving? So what would be the time resolution that you would need to observe molecular dynamics? And here I am borrowing an argument from the Nobel Prize lecture by Ahmed Zewail, who received in 1999 the Nobel Prize in Chemistry for the development of femtochemistry techniques. Now, the argument is very simple, and it says, if you look at a molecule, and you consider the approximate speed at which atoms move within a molecule, these speeds are on the order of 10 to the 3 meters per second. Now, if you consider the typical interatomic distance in a molecule, it's on the order of 1 angstrom, meaning 10 to the minus 10 meters. So it is very easy then to calculate the time resolution. If you want to freeze an atom moving with this speed over a distance of 10 to the 3 meters per second, over a distance of 10 to the minus 10 meter, and you really want to watch the atoms moving within a molecule, you simply need to divide d by v, and you get 10 to the minus 13 seconds. So 100 femtosecond. So you see that the natural time scale for looking at motions of atoms within molecules is the femtosecond time scale. So this is a very simple, but I find very powerful argument. And indeed, if you look at photoinduced processes in molecules, you find a wide range of processes that range from the very early processes that are on the femtosecond time scale. You see 10 to the minus 15 seconds is 1 femtosecond. So you go from tens to hundreds of femtoseconds, and then you have a whole class of longer processes which take place over a longer time scale. But if you want to look at the very fundamental primary events, you need to have femtosecond pulses. Now, how can you generate such short light pulses? And here, indeed, the key advance in ultra-short pulse generation was made possible by the laser. So before the invention of the laser, the shortest man-made light pulse, controllable duration light pulse that could be generated at a duration on the order of 10 nanoseconds. And this was achieved with the so-called Kerr shutter. And still you could perform very valuable experiments with these pulses. And in fact, the Nobel Prize in Chemistry in 1967 was given to Norrish and Porter for the development of flash photolysis. So looking at dynamics over timescales longer than 10 nanoseconds. But when in 1960, the first laser was invented, demonstrated by Ted Nyman, there was immediately a dramatic progress. In fact, in the discovery of mode locking in around the 1965, 1966 allowed already in the late 60s to generate pulses with a duration of maybe 5 picoseconds. So immediately one goes down by 3 orders of magnitude, from a few nanoseconds to a few picoseconds. So you see, you went from microseconds to nanoseconds to picoseconds. And then with the introduction of dye lasers, there was a very rapid progress in the next, say, 20 years, in the following 20 years, which allowed to gain another 3 orders of magnitude. And this is a milestone result in 1988, which was actually performed by Brito Cruz with Chuck Schenck, who managed to generate visible light pulses with a duration of just 6 femtoseconds. So this was a remarkable achievement because this was by far the shortest light pulse ever generated, and it opened up a wide range of investigations. Now, these lasers were dye lasers, which maybe Paolo has worked with them. They are really nasty and difficult to work with. So when you work with this laser, the real experiment is to make the laser work, and only at the end you can really start to do real measurements. But then there was another dramatic progress in the 90s with the introduction of titanium sapphire laser technology. And very soon people started to generate shorter and shorter pulses until, again, this was a milestone result by my colleague Mauro Nisoli at Politecnico. In 1996, one could generate about 4 femtosecond pulses. So using different technologies, either dye lasers or titanium sapphire laser, one can generate pulses with a duration of a few femtoseconds, let's say 4, 5, 6 femtoseconds. So this is, of course, an enormous improvement, a remarkable result. But you may ask yourself, why after such a dramatic progress, then more or less you seem to plateau around durations of a few femtoseconds? So we seem to have reached a fundamental limit. And this is indeed the case if we consider what is a laser pulse, what is a light pulse. So light is an electromagnetic wave that oscillates with the period related to the specific wavelength. So you have the so-called carrier wave, which is a sine wave, which oscillates. And then if you have a pulse of light, you have a few periods of oscillation, maybe 2-3 periods. Now, what is the period? Again, you can make a really simple calculation. You take a red light, for example, like this red pointer, so 600 nanometer, and you calculate easily the period by dividing the wavelength by the speed of light. It's 2 femtoseconds. So visible light has a period on the range of 2 or on the order of 2 femtoseconds. So this means that ultra short light pulses in the visible, which contain a few periods of oscillation of the carrier wave, have a duration of a few femtoseconds. So essentially what we are doing, we are generating pulses which contain only a few cycles of oscillation of the carrier wave, and then they stop. So that's why in my presentation I talk about few optical cycle pulses. And this is the limitation, because an optical pulse must contain at least one cycle, one period of oscillation. So if you are in the visible, when you are generating pulses of a few femtoseconds, you are pretty much close to the limit. Of course, if you want to generate much shorter pulses, you need to go to shorter wavelengths. So if you go at XUV wavelengths, then you can break the femtosecond barrier, and the current limit for generating XUV pulses is on the order of 50 attoseconds. So, but then the wavelength and the period becomes much shorter. Okay, now here, just to give you an idea of how short are these pulses, here I'm making a chart with timescales. So the heartbeat is, let's say, 1 second. So if we consider the human lifetime, it's on the order of 10 to the 9 seconds. If you consider the age of the universe, it's 4.3 10 to the 17 seconds. Now, going with respect to the heartbeat to short times, if you consider the clock of a computer, typical computers are in the gigahertz range, meaning 1 cycle per nanosecond. So we are talking about 10 to the minus 9 seconds. If you consider now the motions of atoms within molecules, which you can study with femtosecond pulses, they are in the range of femtoseconds. Now, if you want to look inside an atom and you want to look at the motion of electron, of the orbital period of electrons within atoms, then you get to even shorter timescale, and then you get to the attosecond timescale. So the attosecond timescale tells you the period of motion of, let's say, electrons within atoms. Now, I want to tell you briefly how to generate an ultra-short light pulse. And to do this, we must understand what are the characteristics of an ultra-short light pulse, which are in a way different from the typical characteristic of a laser. When you think about laser light, if you make an elementary course in lasers, one of the main characteristics that distinguishes laser light is monochromaticity. So lasers typically emit at a very specific color, a very specific wavelength. Now, an ultra-short pulse, by definition, cannot be monochromatic, because it has, having a short duration in time, by the Fourier theorem, it has to contain many frequencies. So you can write the electric field of an ultra-short light pulse in this way, like a carrier wave with a frequency omega 0, which oscillates with this frequency, multiplied by the so-called pulse envelope that tells you the amplitude with respect to the time. Now, if you do the Fourier transform of this light pulse, you get the pulse spectrum, which contains two terms: the so-called spectral amplitude, which tells you how many frequencies, how many different frequency components are contained in your pulse, and the so-called spectral phase. Now, both these terms are crucially important if you want to generate an ultra-short light pulse. So first of all, simply by the Fourier theorem, if you have a short pulse in time, you need to have a broad spectrum. So this is, in a way, you can think of it as an uncertainty principle. So if a pulse is long in time, the envelope has a long duration in time, then the spectrum is narrow. So it has a narrow peaked spectrum. If a pulse is short in time, then the spectrum is broad. This is simply due to the Fourier transform theorem. So a short light pulse, differently from a standard laser pulse, is not monochromatic. Actually, the shorter is the pulse, the broader is its spectrum. So an ideal short pulse would look white because it would contain all the colors, for example, in the visible. So we tend to make pulses, ultra-short pulses, that are as white as possible, that contain as many frequencies as possible, because these pulses will be very short. But it's not enough to have many frequencies. What is very important is also the spectral phase. So the spectral phase is telling me at what is the arrival time of different frequency components of my pulse. So essentially, it's not enough to have many frequencies to get a short pulse. I also need to adjust these frequencies so that they arrive all at the same time. So I need to control also the term, which is the phase. It's like, in a way, it's like having, you know, having to play music. So if you have if you take a symphony of Beethoven, you have a sequence of notes. Now, if you do the Fourier transform of this soundtrack, you will have several peaks, and this is the spectral amplitude. I know that I have this frequency, then this and this, but it's not enough to know which frequencies I have in the music in order to play. I need to know also the time ordering. I need to know when I need to play the different notes. And this is the role of the spectral phase. And if I can adjust properly the spectral phase of my pulse, I will get a very short pulse. So to give you an idea of the role of the spectral phase in a Fourier transform, I like to make this example. Now I'm doing a spatial Fourier transform, but the concept is the same. So I'm taking two pictures. This is the famous Duomo in Milano. It's a very nice, very famous church in my hometown. And this is the Colosseum in Rome, which is also a very famous monument. So now I do the Fourier transform of these images, and I will have a complex term which contains amplitude and phase. So now what I do is I swap, I take the amplitude of the Duomo in Milano and the phase of the Colosseum in Rome, and then I do the inverse Fourier transform. And you see that I get something that is very similar to the Colosseum, and the same is if I do the contrary. So you see that most of the real information is contained in the phase. So in order to generate an ultra-short pulse, I need to control very accurately this term, which is the spectral phase of the pulse. Okay, now I want to tell you very briefly how we generate these pulses. And Paolo mentioned this non-collinear optical parametric amplifier. So the problem is, if I want to generate an ultra-short light pulse, I need to generate a pulse with a broad spectrum. So I need somehow to generate broadband light and also to amplify it. So, and the only way to do this is to exploit nonlinear optical effects. And now I don't have the time to explain in detail all these nonlinear optical effects, but I just want to mention the two key effects. The first effect is so-called femtosecond white light generation. So this is a process that happens when I take a femtosecond pulse, an ultra-short pulse, and I send it into a transparent material. I will get an effect that is known as self-phase modulation. So what happens is that the refractive index of the material is not anymore constant, but it becomes a function of the intensity of light. And in this way, having a pulse that has a time-varying intensity, I get a time-varying refractive index and a time-varying phase. So now, having a time-varying phase, I get an instantaneous frequency, which is the derivative of this phase with respect to time, which will have now some additional components with respect to the carrier frequency. So by doing this, by modulating, the pulse modulates its own phase. That's why it's called self-phase modulation, and it generates new frequencies. So I start from a frequency and I generate frequencies that are lower and higher than the original frequency. And the effect is quite dramatic. So I enter into a transparent material with an infrared pulse that you cannot really see. And thanks to this effect, I generate a whole range of new frequencies. And now my pulse comes out looking white and containing essentially all the frequencies in the visible spectrum. So this is the first effect that we exploit to generate broadband light pulses. Now we need to increase the energy of these pulses. We need to amplify. So a laser is an optical amplifier, but typically a laser works at some specific frequencies. So the second effect that we exploit is called the optical parametric amplifier. And this is a second order nonlinear process. It's a Chi2 effect where the polarization is proportional to the square of the electric field of light. And by using, exploiting this effect, there are many different processes like second harmonic generation, some frequency generation, but I'm particularly interested in this process that is called optical parametric amplification, which is the following process. So I have one pulse, one photon of high energy, which I call the pump photon, which gets absorbed to a virtual level through the optical nonlinearity. Then I come with a lower energy photon, the signal, which stimulates the emission of a second photon from this virtual level. So I now emit a second signal photon, and so I now get two photons out at the output. And then I generate a third photon, which is the idler, which is exactly the difference in frequency between the pump and the signal. So in this way, you see that I am amplifying my signal, and I'm at the same time, in order to conserve the energy, I'm generating a third photon, which is at this different frequency. So this is really an optical amplifier. Now, what is the beautiful about this optical parametric amplifier is that once I fix this frequency, I can now, of course, I need to fulfill energy and momentum conservation, but I can see this OPA as a photon cutter in a way. So I'm taking the energy of this high energy photon, the pump, and I'm cutting it into the sum of two photons, the signal and the idler, with lower energy. Now, the beautiful thing about this concept is that now I am free to do this cut in many different combinations. I can choose many different combinations of signal and idler energies, whose sum gives always the energy of the pump. So I'm free to perform this cutting in many different ways. And so I have automatically, you see, a broadband amplifier. I can amplify many different frequencies. Okay, so this is essentially the concept of ultra broadband optical parametric amplifier, which was pioneered by Christian Manzoni in my department. And you can really apply this concept and generate, using also pulse compression that adjusts the spectral phase. As a result, you can generate really short pulses. Okay, maybe I'm going to skip a couple of technical slides and I'm going to jump to the result. The result is really that you generate really short pulses with a duration of, say, shorter than 10 femtoseconds, and which can be tuned over a very broad range of frequencies, from the visible to the near infrared, and also, as I will show you later, to the ultraviolet. So you see, these are all spectra of ultra-short pulses generated by optical parametric amplifiers that I've generated in my lab, that are tunable from the visible to the near infrared, And all these pulses have been compressed to a duration which is shorter than 10 femtoseconds. And these pulses are all few-optical-cycle pulses. So they contain just a few, maybe 2 or 3 oscillations of the carrier wave. So these are really close to the shortest possible pulse that you can generate at these wavelengths. So for example, this pulse here in the infrared, which has a duration of 8.5 femtoseconds, this pulse is less than 2 cycles of the carrier period. So these are really short pulses. And now you can combine these pulses and you can use them to study very fast processes. Now, what are the experimental techniques once you have generated this pulse to look at, that you can use to look at ultrafast dynamics? Now, there are many different techniques, but the most powerful one, which is used very broadly in my lab and is still the one that I like most because it really has a very broad applicability, is called the pump-probe spectroscopy. So this technique is quite easy to understand. So you can apply it to any sample. It can be a solid, it can be a liquid or a gas, and you have 2 pulses. So the first pulse, which is the pump, is resonantly exciting a system. So the pump pulse promotes from a ground state to an excited state. It can be typically an electronic, but it can also be a vibrational state. It promotes molecules, so it excites a solid. It brings to a photoexcited state the system. And then you come with a second pulse, which is the probe, which is time delayed with respect to the pump, and is typically in a non-collinear configuration so that you can separate after the sample the transmitted probe, and then you have a detector, and this detector measures the so-called differential transmission. So it measures how the transmission of the probe by the sample changes after the sample has been pumped as a function of the delay between pump and probe. So this technique doesn't require you to resolve in time the ultra-short pulses. So this detector is not a fast detector. This detector simply measures the energy of the probe pulse. But what this detector does is it measures it as a function of the delay between pump and probe, which can be controlled with very high precision. Okay, so this is the basic idea. This technique can be used with all kinds of pulses. You can combine terahertz, visible, up to X-rays, and you can choose all possible combinations of pulses as a pump or as a probe to study different phenomena. Now, in order to understand the data that I will show you in the next slides, I just want to discuss the prototypical signals in a pump-probe experiment. Let me assume a 2-level system with a ground state and an excited state. So if I now promote with my pump some molecules from the ground to the excited state, and if I come with a probe that has the same frequency as the pump, I get a photobleaching, because now I have less molecules in the ground state which are able to absorb. So the sample becomes more transparent. It has been bleached. And in this way, I increase the differential transmission signal. So I get an increased transmission, because now I have less molecules which are able to absorb. Now, if I tune my probe, I can, for example, to some vibrational level of the ground state, or even to some different electronic state, I can get stimulated emission. Now I have populated my excited state, and I can stimulate an emission from this state to lower lying state. So this is a real amplification. This is a real optical gain. So it's not a reduced absorption, but it's a real amplification of my probe. Now, the third kind of signal that I can observe is called photoinduced absorption. And now this is an absorption from the excited state that has been populated by the pump pulse to some higher lying state. Now, this is a negative signal, meaning that now having pumped the sample, the absorption of the probe increases. because now I am enabling at this specific wavelength absorption to some higher lying state. So these are the three general signals that you can observe in a typical transient absorption pan-probe experiment, and I will use them also in the following examples. Now, what we do is we are really pushing the time resolution of pan-probe spectroscopy to the 10 femtosecond regime by using two different optical parametric amplifiers that generate pump and probe pulses with different colors. One pulse is used as the pump, and the other pulse is used as the probe. These pulses, the delay of these pulses is controlled by an optical delay line. And what we do after the sample is we disperse the probe, the different colors of the probe on a spectrometer, and we acquire a differential transmission spectrum for a given pan-probe delay. So what we typically get is a map like this. It's a two-dimensional map that contains on one axis the wavelengths, the probe wavelengths, on the other axis the time delay. And so I get essentially for every delay a differential transmission spectrum, or for every wavelength a differential transmission dynamics. So by this two-dimensional map, I can get for every delay a spectrum, and I can get for every wavelength a dynamics. And so this information gives me, this measurement gives me a very rich information about the dynamics of my sample. Now let me come to the second part of my talk. Now I want to use these tools that we have developed to look at important processes that occur in biomolecules. And in particular, I want to look at dynamics through conical intersections. So I want to introduce you briefly to the concept of conical intersections. So typically, when you describe molecular dynamics, you adopt the so-called Born-Oppenheimer approximation, meaning that you separate the electronic and the nuclear coordinates of the wave function. And this approximation solves the Schrödinger equation for electrons for a fixed nuclear position and obtains the electronic energies as a function of those fixed nuclear coordinates, and so obtains the so-called potential energy surfaces. And you can study the wave packet motion along these potential energy surfaces. However, this so-called adiabatic approximation in some cases breaks down, and you have regions in the potential energy landscape where the electronic and the nuclear degrees of freedom become strongly mixed, and this adiabatic approximation breaks down, and these regions are called conical intersections. And these are really regions where the potential energy surfaces, they meet, they become degenerate, and you can think of this region as a doorway through which the photoexcited wave packet can be efficiently funneled, can pass through this door to go to a lower energy electronic state. So these conical intersections are triggering very efficient non-radiative relaxation from one state to another state. Now, this is a representation of a conical intersection. So it's not really a point, it's really a collection of molecular geometries in which these two electronic states become isoenergetic, become degenerate in energy. So this is really a seam, like it's almost a multidimensional line where these surfaces make contact. And so it is really a door for the wave packet to move from one state to another state. For example, here from this state to this state. Now you can distinguish two types of conical intersection. One that is called peaked, where you see that these slopes of the surfaces are opposite. So this kind of conical intersection is promoting so-called photochemistry. So it is promoting the transformation of a molecule from one state, which is called the reactant, to a different state, which is called the product. So you are really doing photochemistry. So you are changing the chemical structure of the molecule, and you go to a product state. So you are triggering with light a chemical reaction. this is called a sloped conical intersection, because now here the slopes of these two curves where they cross are the same. And in this way, what the wave packet does, it cannot go to a proto, but it goes back to the ground state. Okay, it is so this is a way for a molecule to dissipate energy. So this is a way to prevent photochemistry, to prevent a chemical reaction from happening. And now these two types of conical intersections are used by nature to perform two different functions. So these sloped conical intersections are used to perform photochemistry, and in particular, they are used to promote the process of vision. While these sloped conical intersections are used for photoprotection, so to efficiently dissipate the light energy and to prevent the photochemical reaction. Because in DNA, you don't want photochemistry. Photochemistry in DNA is harmful, okay? Now, let me, in the last few minutes of my presentation, let me introduce these two kind of processes. So the first process is the primary event of vision, which is what happens within retinal proteins that are found within our retina. Now, retinal proteins are quite common proteins that are found in many living organisms that have a similar structure that consists of 7 alpha helices. You can count here these 7 helices that form a sort of a pocket, and this pocket encloses the chromophore, which is the molecule responding to visible light, and this chromophore is called the retinal, and this is covalently bound to one of these alpha helices through a covalent bond. And now you can find these retinal proteins either in bacteria, these are called bacterial rhodopsin, or in the visual systems of many higher animals, and these are called visual opsin proteins. Now, in both cases, it's interesting that the photoactivation of this protein occurs through light absorption in the retina, which absorbs visible light, and a photoisomerization. In particular, in so-called bacterial rhodopsin, the retinal is in the all-trans configuration, and it isomerizes to the so-called 13 cis. While in visual rhodopsin, the retinal is in the 11 cis, and it isomerizes to the all-trans. So you see, in both cases, light absorption triggers isomerization. Now, I'm going to focus on visual rhodopsin, which is found in the retina. So if you look at the retina, you find the cones and the rods, and the photoreceptors are contained in the cones that are responsible for color vision, and the rods are responsible for night vision. And in all cases, you have this opsin protein. You have the rhodopsin in the rods and the conopsins in the cones. Now, you see the structure that I showed you before with these 7 ribbons, and here you have the retinal in the 11 cis configuration. Now, what happens when a photon strikes the retina? Here I'm giving you a very schematic 3D representation. So you can see the protein, which is a transmembrane protein. You see the 7 alpha helices represented as cylinders. You see the retinal in the 11 cis. You have a photon striking the retina, the retinal. You have this ultra-fast isomerization from 11 cis to all trans. Now the retinal becomes straight, and now the pocket needs to move to accommodate for the new geometry of the retinal. And you see this cytoplasmic tail that was coiled becomes uncoiled. And this is kind of then triggering an electric signal. So, but essentially the primary event of vision, what really triggers visual perception, is this process of cis-trans isomerization of the retinal within the opsin protein. And this is what we want to study with our experiment, really within the protein. Now, this is, if you look at the photocycle of rhodopsin, it is quite complicated. There is an extremely fast isomerization followed by a whole series of intermediate states, and the result of all is that the retinal will go out of the protein pocket, and so you need an enzyme to restore it back. But okay, now we want to really focus on this very early stage, which again, Professor Brito Cruz was pioneering these studies on bacterial Rhodopsin, but one of the very first experiments after the development of femtosecond laser was performed by Chuck Schenck, was to demonstrate that isomerization of Rhodopsin is an ultra-fast process. And how did they see it? They performed an experiment where they excite the molecule in the excited state of the 11 cis, and then they look at the ground state of the all trans, which has a very characteristic photoinduced absorption, which peaks in the visible. And this famous science paper of 1991 was the really first proof that within 200 femtoseconds, you are already developing this photoinduced absorption, which proves that the wave packet is already in the ground state of the photoproduct, in an all trans. Now, what we really wanted to do in our experiment, with Professor Mattis at Berkeley University, was to really look at the process, at the wave packet moving towards the conical intersection. So what would you need to follow Rhodopsin isomerization in real time? So we come with an ultra-short pulse, we put our wave packet in the excited state of the 11 cis reactant. And now, as the wave packet is in excited state, you can observe a stimulated emission. Now, as the wave packet moves towards the conical intersection, you see that the stimulated emission progressively shifts to lower energies because this gap is becoming narrower. Then you jump, and then you jump through the conical intersection. You are now, after the conical intersection, you are in the ground state of the photoproduct, of the all trans photoproduct. Now you see you cannot emit anymore because you are already on a ground state, but now you can absorb. So the signal changes sign and turns into a photoinduced absorption. And you see that now this photoinduced absorption, the energy will progressively shift to higher energies because now, as this wave packet is relaxing, the band gap is increasing. So there is a very characteristic pattern of stimulated emission shifting to the red, followed by photoinduced absorption shifting to the blue, to higher energies. And we were looking for this characteristic pattern, but this is a very challenging experiment because it requires very high time resolution, so pulses shorter than 20 femtoseconds, and very broad tunability because we want to follow the motion of the wave packet. So let me jump directly to the final result. This was work by Dario Polli in my group. So this is the picture that I would like to show you that really tracks the motion of the wave packet across a conical intersection. Now, here the color code is blue means stimulated emission, red means photoinduced absorption, so negative signal. So here I come, this is a wavelength going from the visible to the near infrared. And here is the time, from time 0. So this experiment has a resolution on the order of 10 femtoseconds. From time 0 up to about 300 femtoseconds. So what happens? Immediately I see a positive signal of stimulated emission. Now this stimulated emission, you see, very quickly shifts to the red. And after about 80 femtoseconds, it disappears because now the wave packet is moving to the conical intersection. So at some point, the band gap becomes so narrow that we cannot follow. But then after we wait, let's say 20, 30 femtoseconds, and then the wave packet reappears. But now the signal is becoming negative because now we have jumped through the conical intersection. We are in the ground state of the photoproduct. And now we are relaxing towards the minimum. And after 200 femtoseconds, we are in this minimum. And then we remain in this minimum essentially for a very long time. Okay? So we are really able to track with this combination of high time resolution and broad spectral coverage. We are able to track the motion of the wave packet through the conical intersection. Now, this started my collaboration with a group of Professor Marco Garavelli at University of Bologna, who was exactly simulating this process, and this required very sophisticated numerical simulation with the so-called QM-MM approach, where you really take the full protein, which is a very big molecule, and you simulate the retinal chromophore, which is the most important part, with a very sophisticated quantum mechanical description. But then you take all the environment of the protein, which is crucial if you want to really understand what's happening, and you describe it with classical, with molecular mechanics. And by this combination of methods, they can achieve a really accurate description of the experiments. So this is the simulation that was performed by the group of Marco Garavelli, where you really see this is simulated emission and photoinduced absorption simulated, and you see there is a really excellent agreement with our experimental results. It's an almost quantitative match with these results. So this is one example of really using ultra-short pulses and the combination of experiments and theory to really get insight into some fundamental biochemical process. So this was done a few years ago, and then I started an effort that lasted for almost 10 years to repeat these experiments on DNA in the ultraviolet. And this is now the very last part of my presentation. And the reason why it was so challenging is that it is very difficult to generate short ultraviolet pulses. Now, very briefly, everybody knows what nucleic acids are. Essentially, DNA is made of a sequence of nucleotides that are composed of a nucleobase, a sugar, and a phosphate group. And you have this three nucleotides essentially bring the information on a protein. And so you're basically encoding proteins in a strand of DNA. Now, of course, the DNA is not made to interact with light. DNA is made to store and preserve information. However, if you look at all the nucleobases that are found both in DNA and in RNA, you will see that they all contain these aromatic rings. And these aromatic rings, these kind of benzene rings, have very strong absorption in the UV. So between, you see, 250 and 300 nanometer. So since there is a lot of UV light in sunlight, the question you may ask is, what happens when you give all this energy, because this energy is on the order of 4 or 5 eV, when you give all this excess energy to a DNA molecule, can it undergo photochemistry? And indeed, this is what happens. So if a DNA strand absorbs UV light, you can have reactions like, for example, the formation of timing dimers. You see here you are breaking this bond, and you are now forming these covalent bonds. And this, of course, is a disruption of the double helix, which creates an error during the copy. This is a so-called genomic lesion. So if you have a lot of these lesions, you can you can have, for example, skin cancer. You can have a lot of, you can damage the genetic code. So why is it possible to preserve DNA despite all the UV light that we have? The reason is that in nucleobases, there are extremely fast and efficient photoprotective mechanisms that prevent the photochemical reactions. So more than 99.9% of the photons are converted in a harmless way on an ultra-fast time scale, so without having time to undergo the photochemistry. So in this case, we are using facendo un seminario in Brasile. Scusa, prova a stare così. So you are, sorry, I had a problem. Okay, now it took me a long time to generate these ultra-short pulses in the ultraviolet, and in the end I managed to generate sub 20 femtosecond ma sono ancora diretto. Just 1 second, sorry, I have a problem. Okay. Sorry, I had a problem. Okay, I hope you can still hear me. Okay, so it took us a long time to generate these sub 20 femtosecond ultraviolet pulses, and so I won't go into the details because I'm already long in time. So essentially, I want to skip to the results. So I'm looking, and I will now look at two of these nucleobases. One is called uridine, which is, or uracil, which is the simplest nucleobase, and the other will be thymidine. So maybe I'm going to skip to the final result, which will be, okay, maybe. So this is now, again, one map looking at the ballistic motion to the conical intersection. So here the color code is, again, blue is a stimulated emission. So we excite the molecule with ultraviolet light to the excited state. Then we get a very fast motion to the conical intersection with the ground state. And so we can observe stimulated emission to the ground state and photoinduced absorption to a higher lying excited state. And this lasts only 100 femtoseconds. After 100 femtoseconds, this disappears. And then I go back to the ground state and I see this photoinduced absorption from the hot ground state. And again, okay, the numerical simulations can reproduce very accurately the experimental results. And just to show you, if we now go to another nucleobase, which is thymidine, again, maybe I can show you. Now this becomes much longer. Now it takes about 1 picosecond for the stimulated emission to disappear. And again, the theory can predict the reason why also this conical intersection is reached in a longer time. So we can really use this very short pulse to look at the primary mechanism with which nucleobases in DNA dissipate the electronic energy and undergo photoprotection mechanism. Okay, so let me skip these points and maybe I can jump to my conclusions. So ultrafast spectroscopy is a powerful technique for the study of primary photoinduced processes in biomolecules. So in this seminar, I tried to give you a general introduction to the topic and also to show you two examples. One is the primary event of vision, which is isomerization of Rhodopsin, and the second is ultrafast photoprotection mechanism in DNA. And with this, I thank you for your attention. Of course, I need to thank my all ultrafast spectroscopy team at Politecnico that really helped me to perform all these experiments. And of course, I want to thank you for your attention.
Thank you very much, Professor Giulio. It was really interesting, very enlightening presentation. I was really amazed about the photodynamics on the retina. Is really interesting. So, so people on YouTube, you are free to make questions. You can type in the chat box of YouTube, and I will read them to Professor Giulio. So I have one. The first question here is Professor Kleber Mendonça. He says, "Thank you, Professor Cerullo, for the excellent talk. Very exciting. So here comes the question. For very short time delays, some coherent artifacts usually appears. how is that managed in your experiments?
Yeah, that's a very good question. Thank you. This is indeed a problem. There are many coherent artifacts that are present at short delays. So actually, there is not really a good way of getting rid of them. So the way that I typically use is I try to minimize all the sources of the coherent artifact. For example, when I work on DNA, I can show you here, we use jets. So we don't use a cuvette because the glass windows of the cuvette could introduce these coherent artifacts. So we are using, you see, a flowing jet. Here you see you have two, the molecules, in this case, they are dissolved in water. They are flown through this jet. So, and then we try to minimize the thickness of this jet and we try also to maximize the concentration of the molecules. But indeed, the problem that you are mentioning is a real one. So we need to fight these coherent artifacts. And sometimes we, of course, can run only the solvent without the molecules and subtract. But this is indeed a problem of these spectroscopies.
Thank you, Professor Giulio. I have another question here from Professor Sergio Muniz. He says, thank you for the nice presentation. And the question is, regarding the last results with DNA, does it means that the thymidine is more prone to UV damage?
Yes.
Is there indication for from the biology supporting this information?
Yeah, this is a very good point, actually. In fact, you have these two molecules, uridine and thymidine, that differ only by a methyl group. And you see that the thymidine has one order of magnitude longer lifetime. So the uridine lives in the excited state only 100 femtosecond, and the thymidine is 1 picosecond. And indeed, thymine dimerization, the formation of thymine dimer, is one of the frequent photolesions of DNA. So, of course, unfortunately, we would like to be able, with our technique, to observe this photo damage, but it's very difficult because this is a quite rare event. So it's not easy to time resolve this process, but indeed, we can correlate the longer excited state lifetime of the thymidine or thymine to the more higher sensitivity to photo damage. This is indeed a problem. This is indeed, but we cannot make a really clear relationship, but this is a very strong indication, at least.
Very interesting. So I will invite Paulo Miranda. Maybe he's co-hosting this presentation with me. Paulo, do you want to make any question to Professor Giulio?
Yes, if I have time, then I would like to ask Giulio, in the example that you showed of the Rhodopsin, the simulations involved the protein pocket and all that.
Yes.
And
yes.
What is the role of this environment in the time dynamics of the isomerization? For instance, if you compare the molecule in solution in just in the buffer, does it change the dynamics?
Yeah, it changes a lot. Thank you, Paolo. This is a very good question. So first of all, yeah, we did the experiment in protein, which is much more difficult than doing in solution. First of all, because you need to get the protein. It needs to be extracted from bovine retinas and purified. But yeah, the point is, if you take the retinal and you put it in solution, the dynamic changes completely. So it becomes much longer. It goes from, let's say, 80 femtosecond to almost a picosecond of excited state lifetime. And also, it goes from, it doesn't have this very efficient, this very high efficiency of isomerization. And the point is, if you compare the protein is very similar to what would happen to the retinal in vacuum. So if you do an experiment on retinal in vacuum, then you would get very similar results to the protein. So the interpretation is that the protein pocket creates an environment which favors these very efficient conical motion to the conical intersection. And the reason is that you, in the case of rhodopsin, you want to be very efficient in going to the photoproduct. So you want to do it as fast as possible because you are linking speed with efficiency. And so in a way, what you're really having is a ballistic motion of the wave packet. So you can really, what I like to think is having this wave packet that is really traveling in a ballistic fashion to the conical intersection and then crossing to the photoproduct. So, and I think this is possible only because of the environment of the protein.
I see. So what you're saying is that probably the solvents, if solvents can get around the molecule, it probably introduced too much fluctuation and this motion becomes hindered, right?
Exactly, yes.
Okay. I have another question. There's no more in the YouTube, no? I have a technical question, Giulio. It's about, you showed in your experiment setups, 2 non-collinear OPAs generating ultra-short laser pulses, one for pumping and another one for probing.
Yes.
And we know that there's another way of doing the probe using the white light continuum, yes, that has a broad spectrum, and therefore, and you can correct for chirp in the probe. Why in these experiments do you need these 2 non-collinear OPAs instead of
that's another very good question. Actually, in general, so the difference, let's say, with the OPA, you can it's quite easy to compress the pulse and to get a really transform limited sub 10 femtosecond. If you use the white light continuum, you can also try to compress it. It's a bit more difficult, but in principle, you are right. I mean, for the pump, of course, we need the short pulse. For the probe, we can also use a chirp, the white light continuum, and still preserve the high time resolution. And in fact, I must say that in the recent in fact, in these experiments that I showed you here on the DNA, now the probe is exactly like you said. I didn't have the time to explain it because I was running out of time. But here you see now the UV probe pulses are made with the white light continuum. And in fact, here you see we have the UV pulse, which is very short. And then we have a white light probe. And then we go to the sample and we get the signal on the spectrometer. So you are perfectly right, Paolo. For the probe, we can avoid the second OPA and just go straight to white light.
Okay. Thank you very much, Giulio. It was a really wonderful talk. Thank you.
Thank you. Thank you very much.
Giulio, very last question. I know you were busy. So my friend, José Paios Neto, he asked if you know any other example in which the biological process is ultra-fast?
Yeah, well, there are many. Well, there are the most important, the essential, another essential biological process that I didn't mention is photosynthesis. Of course, you know, photosynthesis may be even more important because it is the crucial process through which we get all our energy. Even the fossil fuels come from photosynthesis. And if you think in photosynthesis, then you have a sequence of steps. So light is absorbed. You have a very sophisticated machinery in the leaves of essentially chlorophylls and carotenoids. They all absorb energy, and then they are like antennas. So they absorb the energy and they funnel the energy to the so-called reaction center. And the reaction center is where you have the charge separation. And there you have a whole cascade of processes that are ultra fast, from, let's say, tens of femtoseconds to picoseconds. And also for photosynthesis, it is of crucial importance to use ultrafast spectroscopy to really understand these processes. Let's say, I would say the most important are, let's say, photosynthesis, vision, and okay, then photoprotection is not really a photochemical process in the sense that in the case of DNA, you want to avoid photochemistry. but still it's very important to understand how DNA manages to do this. Then there are some additional biomolecules in general that, for example, use light to do like proton pumping or different functions. Another important emerging field is optogenetic, where you can now use light, for example, to open channels and to trigger signals, for example, in neurons and so on.
Fantastic, Professor Giulio. I know you are probably busy, and I think you have to go. So in the name of our institute, I want to thank you very much for your talk and your time here. And hopefully next time we can meet in person, right? Yes,
yes, I hope to. So I really would like to come. And yeah, as I said, I would love to come to Brazil. I've been only once, and I look forward to visit as soon as it will be possible. Hopefully next year will be good for all over the world. It will be possible to go back to a normal life.
There we go.
That
okay, and I would like...
That maybe this is the first colloquium of a long series of Giulio. Thank you.
Hopefully, yes. And I look forward to a churrasco or to a really some good Brazilian food, which I love. You know, fantastic. In Milano, we have a lot of Brazilian restaurants, but I think it's not the same quality as you would have.
So we are going to be waiting you with a good churrasco, picanha churrasco here.
Yes.
So thank you, Giulio, and also Paulo, thank you very much for suggesting the speaker. And just a friendly reminder that we return in 11th of June, talking about teaching in physics and history in physics. Okay, so thank you very much, everyone, and see you in 2 weeks.
Yes.
Bye. See you.